Right now, as you listen to these words, roughly 86 billion neurons are firing in perfect coordination inside your skull. They're processing the sound of this voice, retrieving memories, maintaining your breathing, and somehow generating the very experience of being you. This 3-pound universe tucked between your ears contains more connections than there are stars in the Milky Way. Yet it operates so quietly that most of the time you're completely unaware of its ceaseless work. The human brain represents one of nature's most extraordinary achievements. It's an organ that has pondered the depths of space, composed symphonies, and unlocked the secrets of DNA, all while never directly experiencing the world it so expertly interprets. Every thought you've ever had, every emotion you've felt, every memory you cherish exists because of intricate patterns of electrical and chemical activity flowing through neural networks of staggering complexity. Tonight, we'll journey through these neural landscapes, exploring the architecture of consciousness itself. We'll discover how billions of microscopic cells collaborate to create perception, memory, and thought. We'll visit the ancient structures that keep you alive, and the newer regions that make you uniquely human. As we wander through cortical valleys and subcortical depths, perhaps the gentle rhythm of discovery will guide you toward peaceful rest, your brain continuing its quiet work even as sleep approaches. Wherever you're listening from tonight, and whatever time it is in your corner of the world, I'm delighted you've chosen to spend this time exploring the most fascinating frontier we know. Let's begin our journey into the extraordinary universe within. To understand the brain, we must first appreciate the fortress that protects it, your skull. That familiar dome you carry everywhere represents millions of years of evolutionary refinement in biological armor. Beneath this bony vault lies a delicate organ that controls everything you are, yet remains more fragile than an egg. The central nervous system, comprising the brain and spinal cord, exists in a state of careful isolation from the rest of your body, surrounded by multiple layers of protection that would make a medieval castle designer envious. The outermost guardian is the skull itself, composed of several fused bones that form an almost impenetrable shell. 8 cranial bones create this protective dome, the frontal bone forming your forehead, the parietal bones covering the top and sides, the occipital bone at the back, and the temporal bones housing your ears. These bones fuse completely during childhood, creating seamless armor that can withstand tremendous force. Yet despite this strength, the skull must also accommodate growth, breathing, and circulation through carefully positioned openings and sutures. But bone alone cannot cushion the brain from every impact. Between the skull and brain tissue lie 3 protective membranes called the meninges, each serving a distinct purpose in neural protection. The tough outer layer, the dura mater, clings to the inner skull surface like leather armor. This fibrous membrane creates compartments within the skull, separating different brain regions and preventing excessive movement during head trauma. The middle layer, the arachnoid mater, resembles a delicate spider's web stretched beneath the dura. Its name reflects this gossamer appearance, yet this seemingly fragile membrane plays a crucial role in cerebrospinal fluid circulation. The innermost layer, the pia mater, follows every curve and fold of the brain surface with microscopic precision. Unlike its companions, the pia mater adheres directly to brain tissue, dipping into every sulcus and wrapping around every gyrus. This intimate contact allows blood vessels to penetrate the brain, while maintaining the protective barrier that keeps neural tissue safe from mechanical damage. Together, these 3 membranes create a sophisticated cushioning system that allows the brain to float in protective suspension. Floating within this protective cocoon, the brain bathes in cerebrospinal fluid, a clear liquid that serves as both shock absorber and delivery system. This remarkable fluid circulates continuously through hollow spaces called ventricles, carrying nutrients to brain cells and washing away metabolic waste. The brain contains 4 ventricles, 2 lateral ventricles within the cerebral hemispheres, a 3rd ventricle nestled between deeper structures, and a 4th ventricle connecting to the spinal cord. Each day, your brain produces roughly 500 milliliters of this precious fluid, replacing it entirely 3 or 4 times to maintain the perfect chemical environment for neural function. The cerebrospinal fluid serves purposes beyond simple protection. It maintains constant pressure around the brain, preventing collapse while allowing for slight volume changes during different activities. The fluid also acts as a lymphatic system for the brain, carrying away cellular debris and toxic substances that could interfere with neural function. During sleep, this cleansing process intensifies, washing away proteins associated with neurodegenerative diseases and preparing the brain for another day of conscious activity. The brain itself emerges from three primary divisions during development, each with distinct responsibilities that reflect our evolutionary heritage. The hindbrain, our most ancient neural territory, contains the medulla oblongata, pons, and cerebellum. These structures manage the fundamental business of staying alive: breathing, heart rate, blood pressure, and coordinated movement. The medulla houses the vital centers that control respiration and circulation, operating with such reliability that you never need to consciously remember to breathe or make your heart beat. The pons, meaning bridge in Latin, connects different levels of the nervous system while contributing to sleep regulation and facial sensation. The cerebellum, though, representing only 10% of brain volume, contains over half of all neurons in the nervous system. This remarkable structure fine-tunes movement, maintains balance, and increasingly appears involved in cognitive functions like language and learning. The midbrain, though small, serves as a crucial relay station between higher and lower brain regions. It processes visual and auditory reflexes, controls eye movements, and helps regulate arousal and attention. Within the midbrain lie structures that respond to threats with lightning speed, initiating protective responses before conscious awareness even registers danger. The forebrain represents our newest evolutionary acquisition, housing the massive cerebral hemispheres and deeper structures like the thalamus and hypothalamus. This region generates consciousness, processes complex thoughts, and creates the rich inner world of human experience. The thalamus acts as a relay station for sensory information, while the hypothalamus controls hormones and maintains bodily homeostasis. Within the forebrain's vast territories, we find the capacity for language, abstract reasoning, mathematical thinking, artistic creation, and the ability to contemplate our own existence. Grey matter and white matter paint the brain in distinct shades that reflect their different functions. Grey matter, darker due to the presence of cell bodies and unmyelinated fibers, forms the brain's outer cortex and scattered inner nuclei. Here, neurons make decisions, process information, integrate sensory data, and generate the electrical patterns we call thoughts. The cortical grey matter varies in thickness across different regions, reflecting the computational demands of different brain areas. White matter, pale with fatty myelin sheaths, creates the brain's communication highways. These myelinated axons carry messages between distant regions at speeds approaching 100 meters per second, allowing for the rapid coordination necessary for complex behaviors. The pattern of white matter connections determines much about how the brain processes information, with different connection patterns associated with different cognitive abilities and personality traits. Perhaps the most remarkable feature of brain architecture is the blood-brain barrier, a microscopic gatekeeper that determines what substances can enter neural tissue. The cells lining brain blood vessels fit together so tightly that only specific molecules can pass through, protecting neurons from toxins while allowing essential nutrients like glucose and oxygen to nourish these energy-hungry cells. This barrier transforms the brain into a selective sanctuary, isolated yet connected to the body's circulatory system. Throughout development, the brain follows an intricate choreography of growth and organization that spans decades. Neurons migrate vast distances, measured in cellular terms, to reach their destined locations. They extend long processes called axons and dendrites that seek out specific targets with molecular precision, forming connections guided by chemical signals and genetic programs. This developmental dance creates the basic template upon which experience will write the unique story of each individual brain, establishing the neural foundation for a lifetime of learning, adaptation, and the quiet miracle of consciousness that defines human existence. In the quiet darkness of your brain, a symphony of cellular activity unfolds every moment of your life. Each individual musician in this orchestra is a neuron, a specialized cell evolved over millions of years to carry electrical signals with remarkable precision. These microscopic performers, numbering in the tens of billions, create the neural music that becomes your thoughts, memories, and experiences. Understanding how these cellular virtuosos operate reveals one of nature's most elegant solutions to the challenge of information processing. The neuron begins with its cell body or soma, a roughly spherical structure containing the nucleus and most cellular machinery. This central command center houses the genetic instructions that guide the neuron's behavior, along with the molecular factories that produce proteins essential for neural function. The soma, typically measures between 10 and 25 micrometers in diameter, barely visible to the naked eye, yet containing the complexity necessary to participate in consciousness itself. Extending from the soma, like branches of an ancient tree, dendrites reach outward to receive signals from other neurons. These branching structures create elaborate patterns called dendritic trees, their shapes reflecting the specific computational roles each neuron plays. Some dendrites spread widely to gather information from many sources, while others remain compact and focused. The surface of each dendrite bristles with receptors for neurotransmitters, chemical messengers that convert electrical activity in one neuron into chemical signals that can influence neighboring cells. From the opposite side of the soma emerges the axon, a cable-like projection that can extend extraordinary distances within the brain and beyond. Some axons stretch only micrometers to reach nearby neurons, while others span over a meter to connect the brain with distant muscles. The longest axons in the human body run from the base of the spine to the toes, carrying signals across distances that represent vast journeys in cellular terms. At its origin, the axon emerges from a specialized region called the axon hillock, where the cell integrates all incoming signals and decides whether to generate an action potential. The action potential represents one of biology's most remarkable phenomena, a brief electrical pulse that travels along the axon at speeds up to 100 meters per second. This signal begins when the neuron's membrane potential reaches a critical threshold, triggering the opening of voltage-gated sodium channels. Sodium ions rush into the cell, causing a rapid depolarization that propagates down the axon like a wave. This electrical signal maintains its strength throughout its journey, ensuring that information reaches its destination without degradation. Supporting the neurons in their intricate work are glial cells, often called the brain's unsung heroes. These cellular assistants outnumber neurons by several times and perform essential functions that make neural computation possible. Astrocytes, star-shaped cells that fill the spaces between neurons, regulate the chemical environment around synapses, and help form the blood-brain barrier. They also participate directly in information processing, responding to neural activity and modulating synaptic transmission. Oligodendrocytes perform one of the most crucial support functions by wrapping axons in myelin, a fatty substance that acts like electrical insulation. This myelin sheath dramatically increases the speed of signal transmission while reducing energy consumption. The characteristic white color of myelin gives white matter its name, and allows rapid communication between distant brain regions. Multiple sclerosis, a devastating neurological disease, occurs when the immune system attacks these myelin sheaths, disrupting the brain's communication networks. Microglia serve as the brain's immune system, constantly surveying neural tissue for signs of damage or infection. These cells can rapidly transform from peaceful sentinels into aggressive defenders, engulfing damaged tissue and releasing chemical signals to coordinate protective responses. Recent research suggests that microglia also play important roles in neural development and plasticity, carefully pruning unnecessary connections to optimize brain function. The point where neurons communicate is the synapse, a microscopic gap that represents one of evolution's most sophisticated communication systems. When an action potential reaches the axon terminal, it triggers the release of neurotransmitter molecules stored in tiny vesicles. These chemical messengers drift across the synaptic cleft, a space measuring only 20 to 50 nanometers wide, before binding to receptors on the receiving neuron's membrane. Different neurotransmitters produce different effects on their target neurons. Glutamate, the brain's primary excitatory neurotransmitter, increases the likelihood that the receiving neuron will fire an action potential. GABA, the main inhibitory neurotransmitter, has the opposite effect, making neurons less likely to generate signals. Dopamine modulates motivation and reward, while serotonin influences mood and arousal. Acetylcholine plays crucial roles in attention and muscle control. The strength and efficiency of synaptic connections can change over time through a process called synaptic plasticity. When neurons fire together repeatedly, their connections strengthen, embodying the principle that neurons that fire together wire together. This cellular mechanism underlies learning and memory formation, allowing the brain to adapt its circuitry based on experience. Long-term potentiation, the strengthening of frequently used synapses, creates the neural pathways that encode memories and skills. The brain's remarkable computational power emerges from the intricate patterns of connectivity between billions of neurons. Each neuron typically receives input from thousands of others, and sends its output to thousands more, creating a network of staggering complexity. The human brain contains roughly 100 trillion synapses, each capable of independent modification through experience and learning. The metabolic demands of this neural activity are enormous. Despite representing only 2% of body weight, the brain consumes 20% of the body's energy production. Neurons require constant supplies of glucose and oxygen to maintain their electrical gradients and synthesize neurotransmitters. This high energy demand explains why the brain is so vulnerable to disruptions in blood flow and why consciousness can be lost within seconds when oxygen delivery is interrupted. Different types of neurons specialize in different aspects of information processing. Sensory neurons detect changes in the environment and convert them into electrical signals. Motor neurons control muscle contractions and movement. Interneurons, the most numerous type, process information within the brain and create the complex patterns of activity that generate thoughts and behaviors. Each type has evolved specific structural features that optimize its particular function. Neural plasticity extends throughout life, allowing the adult brain to reorganize its connections in response to new experiences, learning and injury. This remarkable capacity for change means that the neural networks supporting your current thoughts and memories differ significantly from those present in your brain just days or weeks ago. Every experience leaves its mark on neural circuitry, gradually sculpting the patterns of connectivity that define your unique perspective on the world. The silent symphony of neural activity continues uninterrupted throughout life, creating the electrical patterns that emerge as consciousness, emotion and thought. In this moment, as you process these words, millions of neurons are coordinating their activity with precise timing, transforming sensory input into meaning and weaving new connections into the vast tapestry of your brain's neural network. The cerebral cortex represents the crown jewel of human evolution, a thin sheet of neural tissue that transforms our species from clever primates into beings capable of composing poetry, solving mathematical theorems, and contemplating the nature of existence itself. This remarkable structure, barely 3 millimeters thick in most places, contains over 16 billion neurons organized into 6 distinct layers, each performing specialized computational functions that collectively generate the richness of human consciousness. The cortex achieves its extraordinary processing power through an elegant solution to the problem of fitting maximum surface area into the confined space of the skull. Like crumpled paper smoothed and folded to fit within a small box, the cortical surface creates elaborate patterns of ridges and valleys. The raised ridges, called gyri, and the deep valleys, called sulci, increase the brain's surface area nearly threefold compared to a smooth sphere of similar volume. This cortical origami allows the human brain to pack roughly 2.5 square feet of neural real estate into the confines of the cranial vault. The frontal lobe, largest of the cortical regions, stretches from behind your forehead to roughly the crown of your head. This massive expanse of neural tissue houses the most sophisticated aspects of human cognition, including executive control, planning, decision-making, and the ineffable qualities we call personality. The prefrontal cortex, occupying the very front of this region, represents evolution's latest innovation in neural architecture, reaching full maturity only in the mid-20s and remaining vulnerable to age-related decline. Within the frontal lobe lies the primary motor cortex, a strip of tissue that controls voluntary movement throughout the body. This region organizes itself according to a distorted map of the human form called the motor homunculus, where body parts requiring fine control, like the hands and face, receive disproportionately large representation. The adjacent premotor areas plan and coordinate complex movements, transforming abstract intentions into precise sequences of muscle contractions. The frontal lobe also houses Broca's area, typically located in the left hemisphere, which orchestrates the motor aspects of speech production. Damage to this region produces a distinctive pattern of language impairment, where comprehension remains intact but speech becomes labored and telegraphic, demonstrating the specialized nature of cortical function. The frontal regions work tirelessly to integrate information from across the brain, weighing options, inhibiting inappropriate responses, and generating the complex behaviors that define human social interaction. The parietal lobe, positioned behind the frontal region and above the ears, specializes in processing spatial information and creating our sense of bodily awareness. The primary somatosensory cortex, running parallel to the motor strip, receives touch, pressure, and position information from throughout the body. Like its motor counterpart, this region maps the body according to sensitivity rather than actual proportions, with lips and fingertips claiming vast territories compared to the back or legs. Beyond primary sensation, the parietal cortex performs remarkable feats of spatial computation. It integrates information from multiple sensory modalities to create coherent representations of three-dimensional space, tracks the positions of objects and body parts, and guides reaching and grasping movements with millimeter precision. The right parietal lobe plays particularly important roles in spatial attention and awareness, with damage sometimes producing the striking condition of neglect, where patients lose awareness of the left side of space entirely. The parietal regions also contribute to more abstract forms of spatial processing, including mathematical reasoning and the manipulation of mental images. When you rotate an object in your mind's eye or perform mental arithmetic, networks within the parietal cortex spring into action, demonstrating how evolution has repurposed spatial processing mechanisms for higher-order cognition. The temporal lobe, nestled beneath the parietal region and extending toward the ears, serves as the brain's primary hub for auditory processing, language comprehension, and memory formation. The primary auditory cortex receives tonotopically organized input from the ears, creating neural maps that preserve the frequency relationships present in sounds. From these primary areas, auditory information flows into association regions that extract increasingly complex features, ultimately enabling recognition of speech, music, and environmental sounds. The left temporal lobe typically houses Wernicke's area, a region crucial for language comprehension and the selection of appropriate words and meanings. Unlike Broca's area, damage to Wernicke's region produces fluent but meaningless speech, highlighting the distributed nature of language processing across multiple cortical networks. Deep within the temporal lobe lies the hippocampus, a seahorse-shaped structure essential for forming new explicit memories. This remarkable region creates cognitive maps of both physical and conceptual spaces, binding together the diverse elements of experience into coherent episodes that can be consciously recalled. The surrounding temporal cortex stores these memories in distributed networks, with different regions specializing in different categories of information. The occipital lobe, smallest of the cortical regions, occupies the back of the brain and dedicates itself almost entirely to visual processing. The primary visual vortex receives input from the eyes, organized according to spatial location, creating retinotopic maps that preserve the geometric relationships present in the visual world. From this foundation, visual information flows through two major processing streams. The ventral stream, extending into the temporal lobe, specializes in object recognition and answers the question "what?", while the dorsal stream, projecting into the parietal lobe, focuses on spatial relationships and motion, addressing where and how. The remarkable specificity of visual processing becomes apparent when examining the response properties of individual neurons. Some cells respond only to horizontal lines, others to vertical orientations, and still others to specific directions of motion. Higher-level areas contain neurons selective for faces, places, or even specific individuals, demonstrating how the cortex builds complex representations from simple visual features. Early anatomists divided the cortex into 52 distinct areas based on cellular architecture, a classification system developed by Corbinian Brodmann that remains influential today. Each Brodmann area exhibits characteristic patterns of neuronal organization, reflecting specialized functions that have been refined through millions of years of evolution. Modern neuroscience has revealed that these anatomical divisions correspond remarkably well to functional specializations identified through contemporary brain imaging techniques. The corpus callosum, a massive bundle of over 200 million axons, connects the left and right cerebral hemispheres, allowing information to flow freely between cortical regions. This neural bridge enables the unified conscious experience despite the brain's bilateral organization. Studies of patients with severed corpus callosum connections have revealed fascinating asymmetries in hemispheric function, with the left hemisphere typically specializing in language and analytical processing, while the right hemisphere excels at spatial and holistic tasks. However, the popular notion of left brain versus right brain personality types oversimplifies the elegant cooperation between hemispheres. In healthy brains, both sides work together seamlessly, with the corpus callosum ensuring that information processed in one hemisphere becomes available to the other within milliseconds. This interhemispheric communication allows for the integration of different types of processing, combining analytical and intuitive approaches to create the full spectrum of human cognition. The cortical layers themselves represent distinct computational stages, with information flowing in characteristic patterns through the laminar architecture. Sensory input typically arrives in layer 4, spreads to layers 2 and 3 for local processing, and then projects to layer 5 for output to other brain regions. Layer 6 provides feedback to the thalamus, creating loops that modulate incoming sensory information based on current brain states and expectations. This laminar organization, repeated across all cortical areas with subtle variations, suggests that the cortex employs common computational principles despite its functional diversity. Recent theories propose that cortical circuits perform predictive coding, constantly generating expectations about incoming information and updating these predictions based on sensory input. This perspective transforms our understanding of perception from passive reception to active construction, where the brain continuously creates models of the world and refines them through experience. The cortical surface area varies considerably among individuals, with larger brains generally containing more cortical real estate. However, intelligence correlates more strongly with cortical thickness and the efficiency of neural connections than with overall brain size. The human cortex continues to develop throughout childhood and adolescence, with different regions maturing at different rates, explaining why abstract reasoning abilities and impulse control develop gradually over the first 2 decades of life. Beneath the elaborate folds of the cerebral cortex lies a collection of ancient structures that have guided animal behavior for hundreds of millions of years. These subcortical regions represent the brain's oldest territories, regions that emerged when our distant ancestors first developed primitive nervous systems. While the cortex contemplates philosophy and composes symphonies, these deeper structures quietly manage the fundamental business of staying alive, regulating heartbeat, breathing, hormone production, and the basic drives that ensure survival and reproduction. The brainstem forms the most primitive foundation of the human brain, connecting the cerebral hemispheres to the spinal cord, like the trunk of an ancient tree. This vital structure contains 3 major divisions, each evolved at different points in vertebrate history to serve essential life-sustaining functions. The medulla oblongata, the most caudal portion, houses the cardiovascular and respiratory control centers that maintain life from moment to moment. Within its compact neural circuits lie the neurons that regulate heart rate, blood pressure, and breathing rhythm, with such reliability that these functions continue even during the deepest stages of sleep. The medulla also controls reflexive behaviors like swallowing, coughing, and vomiting, protective mechanisms that prevent aspiration and expel harmful substances. Damage to this region proves rapidly fatal, demonstrating the critical importance of these ancient neural circuits. The medulla's respiratory centers automatically adjust breathing rate and depth based on carbon dioxide levels in the blood, maintaining the delicate chemical balance necessary for cellular function throughout the body. Above the medulla lies the pons, named for its bridge-like appearance, as it connects different levels of the nervous system. This structure contributes to sleep regulation, facial sensation, and eye movement control. The pons houses nuclei that generate rapid eye movement sleep, the stage during which most vivid dreaming occurs. It also contains centers that coordinate the complex muscular activities involved in chewing, facial expression, and the precise eye movements that track objects through space. The midbrain, though small, serves crucial functions in sensory processing and motor control. The superior colliculus processes visual information and controls reflexive eye movements, allowing rapid orientation towards sudden visual stimuli. The inferior colliculus handles similar functions for auditory information, enabling quick responses to unexpected sounds. These structures operate with millisecond precision, initiating protective responses before conscious awareness even registers potential threats. Deep within the midbrain lies the substantia nigra, a darkly pigmented region that produces dopamine for movement control and reward processing. The death of neurons in this area causes Parkinson's disease, demonstrating how the loss of these ancient cells can profoundly impact modern human experience. The midbrain also contains the red nucleus, which contributes to motor coordination, and the periaqueductal gray, a region involved in pain modulation and defensive behaviors. The thalamus occupies a central position both anatomically and functionally, serving as the brain's primary relay station for sensory information. This egg-shaped structure consists of numerous nuclei, each specialized for processing specific types of information before forwarding it to appropriate cortical regions. Visual information from the eyes passes through the lateral geniculate nucleus, auditory signals traverse the medial geniculate nucleus, and touch sensations relay through the ventral posterior nucleus. Beyond simple relay functions, the thalamus actively filters and modulates sensory information based on current behavioral states and attention. During sleep, thalamic circuits generate the rhythmic oscillations that characterize different sleep stages, while damage to specific thalamic nuclei can produce profound alterations in consciousness and awareness. The thalamus also participates in motor control through its connections with the basal ganglia and cerebellum, demonstrating the integrated nature of brain function. The hypothalamus, despite weighing only 4 grams, exerts enormous influence over bodily functions through its control of the endocrine system. This remarkable structure contains nuclei that regulate body temperature, hunger, thirst, circadian rhythms, and reproductive behavior. The hypothalamus monitors internal conditions through specialized neurons that detect changes in blood chemistry, temperature, and osmolarity, responding by adjusting hormone production and triggering appropriate behavioral responses. The suprachiasmatic nucleus within the hypothalamus serves as the body's master clock, receiving direct input from the eyes about light levels and synchronizing circadian rhythms throughout the body. The paraventricular and supraoptic nuclei produce hormones like vasopressin and oxytocin, which regulate water balance, social bonding, and childbirth. The arcuate nucleus monitors nutrient levels and produces signals that control appetite and metabolism, playing crucial roles in maintaining energy homeostasis. The limbic system encompasses a collection of interconnected structures that process emotions, motivations, and memories. The hippocampus, perhaps the most studied component, creates cognitive maps of both physical and conceptual spaces while binding together the elements of experience into episodic memories. This seahorse-shaped structure contains place cells that fire when animals occupy specific locations, providing the neural foundation for spatial navigation and autobiographical memory. The amygdala, an almond-shaped cluster of nuclei, specializes in processing fear, threat detection, and emotional learning. This ancient structure can trigger fight-or-flight responses within milliseconds of detecting potential danger, often before conscious awareness recognizes the threat. The amygdala also modulates memory formation, ensuring that emotionally significant events receive preferential encoding and storage. Different amygdala nuclei contribute to various aspects of emotional processing, from fear conditioning to social recognition. The cingulate cortex wraps around the corpus callosum like a collar, integrating emotional and cognitive information, while monitoring conflicts between competing response tendencies. The anterior cingulate cortex activates during physical pain, emotional distress, and cognitive conflicts, suggesting that these diverse experiences share common neural mechanisms. The posterior cingulate cortex contributes to self-referential thinking and the default mode network that activates during rest and introspection. The basal ganglia form a complex circuit that refines movement and helps establish habitual behaviors. This collection of nuclei, including the caudate nucleus, putamen, and globus pallidus, receives input from across the cortex and sends processed information back through the thalamus. The basal ganglia circuit operates like a sophisticated filtering system, facilitating desired movements while suppressing unwanted motor activity. Different regions within the basal ganglia contribute to distinct aspects of behavior. The dorsal striatum, comprising the caudate and putamen, processes goal-directed actions and motor learning. The ventral striatum, including the nucleus accumbens, responds to rewards and motivational stimuli, playing crucial roles in addiction and decision-making. Dysfunction in basal ganglia circuits underlies movement disorders like Parkinson's disease and Huntington's disease, as well as psychiatric conditions involving compulsive behaviors. The septum, fornix, and mammillary bodies complete the limbic circuit, providing additional connections between memory and emotional systems. The septal nuclei modulate hippocampal activity and contribute to social behaviors, while the fornix serves as a major output pathway from the hippocampus. The mammillary bodies, though small, play important roles in memory consolidation and spatial navigation. These subcortical structures work in constant coordination with cortical regions, creating the integrated neural networks that generate behavior and experience. While the cortex provides sophisticated analysis and planning capabilities, these ancient structures ensure that basic survival needs remain prioritized and that learned behaviors become automatic through repetition. The seamless cooperation between old and new brain regions demonstrates evolution's tendency to build upon existing foundations rather than starting fresh, creating the layered architecture that characterizes the human brain. The rhythmic activities generated by these deep structures continue throughout life, maintaining the biological functions that support conscious experience while remaining largely outside awareness. In sleep, these circuits continue their essential work, regulating breathing and circulation while orchestrating the neural activities that consolidate memories and prepare the brain for another day of conscious engagement with the world. The brain exists in profound isolation, sealed within the protective fortress of the skull and never directly experiencing the world it so expertly interprets. Every sensation you've ever felt, every color you've perceived, every sound that has moved you to tears, exists only as patterns of electrical activity flowing through neural pathways. The transformation of physical energy into conscious experience represents one of nature's most remarkable achievements, accomplished through specialized cellular gateways that convert light waves, sound vibrations, and molecular interactions into the language of neurons. Visual information begins its journey at the retina, a thin sheet of neural tissue lining the back of each eye. Here, photoreceptor cells called rods and cones capture photons and convert light energy into electrical signals through a cascade of molecular reactions. Rods, numbering roughly 120 million per eye, respond to dim light and provide our night vision, while 6 million cones detect color and fine detail during daylight conditions. These photoreceptors synapse with bipolar cells, which in turn connect to ganglion cells whose axons form the optic nerve carrying visual information toward the brain. The optic nerves from each eye partially cross at the optic chiasm, ensuring that information from both eyes reaches both hemispheres while maintaining the precise spatial relationships present in the visual world. Visual signals first reach the lateral geniculate nucleus of the thalamus, which organizes information into parallel streams before forwarding it to the primary visual cortex in the occipital lobe. This initial cortical processing area contains neurons arranged in columns that respond to specific features like orientation, direction of motion, and eye dominance. From primary visual cortex, information flows along two major pathways that extract different aspects of visual experience. The ventral stream, projecting toward the temporal lobe, specializes in object recognition and identification, answering the fundamental question of what we are seeing. Neurons along this pathway respond increasingly specifically as information progresses, with some cells responding only to faces, others to specific objects, and still others to particular individuals. The dorsal stream, extending into the parietal lobe, processes spatial relationships and motion, determining where objects are located and how they move through space. Auditory processing begins when sound waves cause the eardrum to vibrate, transmitting mechanical energy through the tiny bones of the middle ear to the fluid-filled cutera of the inner ear. Within the cutera, hair cells detect these vibrations and convert them into electrical signals that preserve the frequency content of the original sound. Different frequencies activate hair cells at different locations along the cutera spiral, creating a tonotopic map that maintains frequency relationships throughout the auditory pathway. Auditory nerve fibers carry these signals to the cutera nuclei in the brainstem, where initial processing separates information into parallel streams. Some pathways specialize in detecting the timing differences between sounds reaching each ear, crucial for localizing sound sources in space. Others focus on intensity differences or spectral content, extracting features necessary for speech recognition and environmental awareness. These processed signals converge in the inferior colliculus of the midbrain before reaching the medial geniculate nucleus of the thalamus, and ultimately the primary auditory cortex in the temporal lobe. The somatosensory system creates our sense of touch, pressure, temperature, and body position through an elaborate network of specialized receptors distributed throughout the skin, muscles, and joints. Different receptor types respond to specific mechanical stimuli. Meissner's corpuscles detect light touch and low-frequency vibrations. Pacinian corpuscles sense deep pressure and high-frequency vibrations. Ruffini endings respond to skin stretch and contribute to position sense, while free nerve endings detect pain, temperature, and crude touch sensations. These diverse sensory signals travel through peripheral nerves to the spinal cord, where they ascend in organized pathways that preserve the spatial relationships of the body surface. The dorsal column pathway carries fine touch and position information directly to the medulla, while the spinothalamic tract transmits pain and temperature signals with multiple synaptic stops along the way. Both pathways ultimately reach the ventral posterior nucleus of the thalamus before projecting to the primary somatosensory cortex, which contains a distorted map of the body surface that reflects the density of sensory innervation rather than actual body proportions. Olfactory processing follows a unique pathway that bypasses the thalamus entirely, providing the most direct sensory connection to the brain. Odor molecules dissolve in the mucus lining the nasal cavity and bind to receptors on olfactory sensory neurons, each expressing only one type of odor receptor from a family of roughly 400 different receptor genes. These neurons project directly to the olfactory bulb, where their axons converge in spherical structures called glomeruli, according to receptor type. From the olfactory bulb, processed odor information travels along multiple pathways to different brain regions. Some projections reach the piriform cortex for conscious odor perception, while others connect to the amygdala for emotional responses and to the hippocampus for memory formation. This direct connection to limbic structures explains why smells can trigger such powerful emotional memories, and why olfactory experiences often feel more immediate and visceral than other sensory modalities. Gustatory processing begins with taste buds distributed across the tongue, soft palate, and throat, each containing specialized cells that respond to one of 5 basic taste qualities: sweet, sour, salty, bitter, and umami. These taste receptor cells detect dissolved chemicals and transmit signals through 3 cranial nerves to the brainstem gustatory nuclei. Taste information then ascends to the ventral posterior medial nucleus of the thalamus, before reaching the primary gustatory cortex in the insula and frontal operculum. Pain processing involves a complex network of pathways that transform potentially harmful stimuli into the conscious experience of pain while simultaneously triggering protective responses. Nociceptors, specialized nerve endings that detect tissue damage, respond to mechanical, thermal, or chemical stimuli that threaten bodily integrity. These signals travel through 2 main pathways: fast myelinated fibers that produce sharp, localized pain, and slower unmyelinated fibers that generate dull, aching sensations. Pain signals undergo extensive processing and modulation as they ascend through the nervous system. The spinal cord contains circuits that can amplify or suppress pain signals based on concurrent input from other sensory modalities, explaining phenomena like the ability of rubbing an injury to reduce pain sensation. Descending pathways from the brain can also modulate pain perception, allowing psychological factors like attention, expectation, and emotional state to profoundly influence pain experience. The brain integrates information from multiple sensory modalities to create unified perceptual experiences through a process called multisensory binding. Neurons in association areas respond to combinations of sensory inputs, enabling the brain to determine which signals originate from the same external source. This integration occurs automatically and largely outside conscious awareness, creating the seamless sensory experience that characterizes normal perception. Sensory adaptation represents another crucial aspect of neural processing, allowing the brain to adjust its sensitivity based on the current sensory environment. Neurons gradually reduce their response to constant stimuli while maintaining sensitivity to changes, ensuring that the brain remains responsive to new information while avoiding saturation from unchanging background conditions. This adaptation occurs at multiple levels, from individual receptor cells to complex cortical circuits. The brain's predictive processing capabilities transform sensory perception from passive reception to active construction. Rather than simply responding to incoming stimuli, the brain constantly generates predictions about expected sensory input based on prior experience and current context. These predictions influence how sensory information is processed and interpreted, explaining phenomena like perceptual illusions and the role of expectation in shaping conscious experience. Each sensory system contributes unique information about the external world, while sharing common organizational principles that reflect the brain's evolutionary heritage. The transformation of physical energy into neural activity and ultimately conscious experience represents one of the most profound mysteries in neuroscience, bridging the gap between objective physical events and subjective phenomenal experience through the elegant mechanisms of sensory transduction and neural processing. Memory represents the brain's most mysterious and essential function, transforming fleeting experiences into enduring traces that shape our understanding of the world and ourselves. Without memory, consciousness would exist only in an eternal present, unable to learn from the past or plan for the future. The neural mechanisms that allow a 3-pound organ to store decades of experiences, from childhood birthday parties to professional skills, involve some of the most sophisticated biological processes evolution has ever produced. The brain contains multiple memory systems, each specialized for different types of information and operating according to distinct principles. Explicit memory, also called declarative memory, encompasses the conscious recollection of facts and events that can be verbally described or mentally visualized. This system divides into episodic memory, which stores personal experiences tied to specific times and places, and semantic memory, which contains general knowledge about the world, independent of when or where it was learned. Implicit memory operates outside conscious awareness, influencing behavior through learned associations and skills that resist verbal description. Procedural memory enables the acquisition and retention of motor skills like riding a bicycle or playing a musical instrument, creating automated patterns of movement that improve with practice. Priming effects demonstrate how previous exposure to stimuli influences subsequent processing, while conditioning mechanisms associate neutral stimuli with meaningful outcomes through repeated pairings. The encoding process transforms sensory experiences into neural representations that can be stored and later retrieved. This transformation requires attention, as information outside the focus of awareness rarely forms lasting memories. The brain selectively filters incoming information based on novelty, emotional significance, and relevance to current goals, ensuring that memory resources are allocated to experiences most likely to influence future behavior. During encoding, the hippocampus serves as a temporary binding site that links together the deterred cortical representations of an experience. Rather than storing memories itself, the hippocampus creates indices that point to the various cortical regions where different aspects of an experience are represented. Visual details might be stored in visual cortex, auditory components in auditory regions, and emotional aspects in the amygdala, with the hippocampus maintaining the connections between these distributed elements. The consolidation process gradually transfers memories from hippocampal to cortical storage, a transformation that can take months or years to complete. During this period, memories remain vulnerable to disruption, explaining why recent memories are more susceptible to loss following brain injury than older, well-consolidated recollections. Sleep plays a crucial role in consolidation, with slow-wave sleep facilitating the transfer of information from temporary hippocampal storage to permanent cortical networks. Long-term potentiation represents the cellular mechanism underlying learning and memory formation. When neurons fire together repeatedly, the synaptic connections between them strengthen through changes in receptor sensitivity and the growth of new synaptic contacts. This process embodies the principle that neurons that fire together wire together, creating the physical changes in brain connectivity that encode learned associations and skills. The molecular machinery of memory involves complex cascades of gene expression and protein synthesis that modify synaptic structure and function. Immediate early genes like CFOS activate within minutes of learning experiences, initiating the production of proteins necessary for long-term synaptic changes. These molecular events transform temporary electrical activity into permanent structural modifications that can persist for decades. Working memory provides a mental workspace where information can be temporarily held and manipulated during cognitive tasks. The prefrontal cortex orchestrates this system, maintaining active representations of goal-relevant information while filtering out distractions. Working memory capacity limits constrain the amount of information that can be simultaneously processed, explaining why complex mental tasks become difficult when too many elements must be juggled at once. Different regions of the prefrontal cortex contribute specialized functions to working memory operations. The dorsolateral prefrontal cortex maintains and manipulates spatial and temporal information, while the ventrolateral prefrontal cortex processes object features and verbal material. The anterior cingulate cortex monitors conflicts between competing information sources and signals when additional cognitive control is needed. Memory retrieval involves reconstructing past experiences from distributed neural traces, rather than simply replaying stored recordings. This reconstructive process makes memory inherently malleable, as each act of remembering can modify the neural representation being retrieved. The context in which retrieval occurs influences what aspects of a memory are accessed, explaining why returning to the location where something was learned can facilitate recall. The phenomenon of reconsolidation reveals that even well-established memories become temporarily labile when retrieved, requiring renewed consolidation to maintain their stability. This process provides opportunities for memory updating and modification, but also creates vulnerabilities where retrieved memories can be altered or disrupted. Therapeutic approaches for traumatic memories increasingly target these reconsolidation windows to reduce the emotional impact of distressing recollections. Forgetting represents an active process rather than simple decay, involving neural mechanisms that deliberately weaken or eliminate memory traces. Forgetting serves adaptive functions by preventing interference from outdated information and reducing the cognitive burden of maintaining irrelevant memories. The brain appears to selectively retain information that remains relevant while allowing unused memories to fade through disuse and active suppression. Sleep profoundly influences memory consolidation through mechanisms that operate during different sleep stages. Slow-wave sleep facilitates the transfer of hippocampal memories to cortical storage sites, while rapid eye movement sleep appears particularly important for procedural learning and emotional memory processing. The sleeping brain replays patterns of neural activity that occurred during waking experiences, strengthening the synaptic connections that encode these memories. Memory disorders reveal the intricate organization of the brain's memory systems through the patterns of preserved and impaired function they produce. Alzheimer's disease initially affects the hippocampus and associated medial temporal lobe structures, producing profound deficits in new learning while leaving procedural skills and remote memories relatively intact. Korsakoff's syndrome, resulting from chronic alcohol abuse and thiamine deficiency, creates severe anterograde amnesia, while sparing working memory and implicit learning abilities. The emotional modulation of memory demonstrates how the amygdala and associated limbic structures influence the strength and durability of memory formation. Emotionally arousing experiences receive preferential encoding and consolidation, explaining why traumatic events and peak positive experiences often form the most vivid and persistent memories. Stress hormones like cortisol can enhance memory consolidation at moderate levels, but impair both encoding and retrieval when present in excessive amounts. Memory development follows a protracted course that extends well into adolescence, reflecting the gradual maturation of hippocampal and prefrontal circuits. Infantile amnesia, the inability to recall experiences from early childhood, results from the immaturity of memory systems rather than simply the passage of time. As these systems mature, children develop increasingly sophisticated abilities to encode, organize, and retrieve episodic memories. The social dimensions of memory reveal how cultural and interpersonal factors shape both what we remember and how we remember it. Collaborative remembering, where groups work together to reconstruct shared experiences, can both enhance accuracy through the pooling of complementary details and introduce errors through social influence and suggestion. The stories we tell about our past experiences become part of the memories themselves, illustrating the dynamic relationship between memory and narrative identity. False memories demonstrate the reconstructive nature of recollection, showing how confident memories can form for events that never occurred. These false recollections often arise from source confusion, where details from different experiences become merged, or from suggestion, where external information becomes incorporated into personal memories. The malleability of memory has important implications for eyewitness testimony and therapeutic practices that involve memory recovery. Recent research has revealed that memories exist in multiple trace states, with some becoming increasingly independent of the hippocampus over time, while others retain hippocampal dependence indefinitely. This multiple trace theory suggests that detailed episodic memories always require hippocampal involvement, while general semantic knowledge can eventually become hippocampus independent. The transformation of episodic memories into semantic knowledge represents one of memory's most important functions, allowing specific experiences to contribute to general understanding. Language represents humanity's most distinctive achievement, a complex communication system that sets our species apart from all others on Earth. While many animals possess sophisticated methods of conveying information, only humans have developed the capacity to generate infinite novel expressions from finite elements, creating meaning through the recursive combination of sounds, words, and grammatical structures. The neural networks that enable language processing span multiple brain regions, working together to transform thoughts into speech and decode the utterances of others into comprehensible meaning. The classical model of language organization identified two primary cortical regions, Broca's area in the left frontal lobe and Wernicke's area in the left temporal lobe. Paul Broca first described his eponymous area in 1861, after observing patients who could understand speech, but struggled to produce fluent language. These individuals, suffering from what became known as Broca's aphasia, retained comprehension abilities while exhibiting labored telegraphic speech patterns that preserved meaning while omitting grammatical elements. Carl Wernicke later identified a temporal lobe region, whose damage produced a different pattern of language impairment. Patients with Wernicke's aphasia produced fluent but meaningless speech, demonstrating intact grammar and prosody while lacking semantic content. Their comprehension also suffered, creating a profound disconnect between the form and meaning of language. This double dissociation between production and comprehension deficits suggested that different brain regions specialized for distinct aspects of language processing. Modern neuroscience has revealed that language processing involves far more extensive networks than the classical Broca-Winnicke model suggested. Contemporary research identifies multiple streams of language processing that operate in parallel, connecting frontal, temporal, and parietal regions through white matter pathways. The dorsal stream links temporal and frontal regions through the arcuate fasciculus, supporting the mapping between sounds and articulation while processing grammatical relationships. The ventral stream connects temporal regions with inferior frontal areas through the uncinate fasciculus, emphasizing semantic processing and conceptual understanding. Speech production requires extraordinary coordination between cognitive planning and motor execution. The process begins in conceptual areas where speakers formulate the ideas they wish to express. These concepts architect semantic networks that retrieve appropriate words from long-term memory, involving regions throughout the left temporal lobe that store different categories of knowledge. The inferior frontal gyrus, including Broca's area, orchestrates the selection of specific words while suppressing competing alternatives. Phonological processing transforms selected words into sequences of sounds through networks in the left hemisphere that map between orthographic, phonological, and motor representations. The superior temporal sulcus processes the temporal patterns of speech sounds, while the inferior parietal lobe integrates auditory and motor information necessary for speech production. The primary motor cortex and associated premotor areas execute the precise muscular movements required to shape the vocal tract and produce intelligible speech. Reading and writing represent relatively recent cultural innovations that piggyback on pre-existing neural circuits evolved for spoken language. The visual word form area in the left fusiform gyrus responds specifically to letter strings and written words, connecting visual processing with phonological and semantic networks. Skilled readers develop automatic connections between orthographic patterns and their corresponding sounds and meanings, enabling rapid text comprehension without conscious effort. The neural pathways supporting reading vary depending on the writing system being processed. Alphabetic scripts like English rely heavily on phonological processing, activating brain regions that map between letters and sounds. Logographic scripts like Chinese emphasize visual semantic processing, engaging different networks that connect character recognition directly with meaning. Bilingual individuals who read multiple writing systems show remarkable flexibility in switching between these different processing modes. Bilingual brain organization reveals the plastic nature of language networks and their capacity to accommodate multiple communication systems. Early bilinguals who acquire two languages simultaneously often show overlapping neural representations with similar brain regions supporting both languages. Late bilinguals who learn a second language after childhood typically develop more distinct neural networks, with additional activation in frontal control regions that manage competition between languages. The degree of neural overlap over bent languages depends on their linguistic similarity and the age of acquisition. Languages that share similar grammatical structures and sound systems show greater neural convergence than those with distinct organizational principles. Proficiency also influences brain organization, with more fluent speakers showing greater neural efficiency and reduced activation in control regions during language switching. Language development follows a predictable sequence that reflects the gradual maturation of underlying neural systems. Infants begin life with the capacity to discriminate speech sounds from all human languages, but this universal sensitivity narrows during the first year as exposure shapes neural tuning to the specific sounds of the native language. The critical period for language acquisition extends through early childhood, during which the brain remains maximally plastic for learning linguistic rules and patterns. Phonological development precedes semantic and grammatical acquisition, with children first mastering the sound patterns of their native language before developing sophisticated vocabulary and syntactic abilities. The left hemisphere language regions show progressive specialization throughout childhood and adolescence, with some aspects of language processing continuing to mature into the teenage years. Sign languages provide compelling evidence for the independence of language from the auditory vocal channel. Deaf individuals who use sign languages show activation in the same left hemisphere regions that support spoken language, including Broca's and Wernicke's areas. This demonstrates that these brain regions respond to linguistic structure rather than specific sensory modalities. supporting the abstract nature of language processing. The spatial properties of sign languages engage additional neural resources compared to spoken languages, particularly in the right hemisphere regions that process spatial relationships. However, the core language networks remain left lateralized, showing that the fundamental organization of language processing transcends the specific modality used for expression. The relationship between thought and language remains one of the most debated topics in cognitive science. Some theories propose that language shapes thought by providing conceptual categories that influence how we perceive and understand the world. Other perspectives argue that thought is largely independent of language, with linguistic expression serving primarily as a tool for communication rather than cognition. Neuroimaging studies reveal that many cognitive processes operate independently of language networks, suggesting that thought can occur without linguistic mediation. However, language appears to facilitate certain types of abstract reasoning, and may influence how information is organized and recalled from memory. The interaction between linguistic and non-linguistic cognition likely varies depending on the specific cognitive domain and the individual's linguistic experience. Language disorders illuminate the intricate organization of linguistic knowledge in the brain. Primary progressive aphasia causes gradual deterioration of language abilities while initially sparing other cognitive functions, demonstrating the relative independence of language networks. Different variants of this condition affect distinct aspects of language processing, with semantic variant primarily impacting word meaning, logopenic variant affecting phonological processing, and non-fluent variant disrupting grammatical abilities. The remarkable recovery that can occur following language disorders reveals the plastic nature of neural language networks. Speech therapy interventions can promote reorganization of damaged circuits or recruitment of alternative pathways to restore communicative abilities. The extent of recovery depends on factors including the location and size of brain damage, the age at onset, and the intensity of rehabilitation efforts. Consciousness represents perhaps the greatest mystery in neuroscience, the seemingly impossible transformation of electrical and chemical activity into subjective experience. How does the firing of neurons give rise to the felt sense of being you, the inner movie of awareness that accompanies every waking moment? Despite decades of research, the mechanisms that generate consciousness from neural activity remain largely enigmatic, though modern neuroscience has begun to identify the brain networks and processes that correlate with different states of awareness. The neural correlates of consciousness encompass the minimal set of brain mechanisms sufficient to produce specific conscious experiences. These correlates differ depending on the content of consciousness, with visual awareness involving occipital and temporal cortices, auditory consciousness engaging temporal regions, and bodily awareness activating somatosensory areas. However, consciousness also requires more general mechanisms that integrate information across sensory modalities and maintain the unified field of awareness that characterizes normal waking experience. The global workspace theory proposes that consciousness arises when information becomes globally available across multiple brain networks through widespread cortical broadcasting. According to this model, local processing occurs unconsciously in specialized brain regions until competitive processes select certain information for global distribution. Once broadcast globally, this information becomes consciously accessible and can influence decision-making, memory formation, and verbal report. Neuroimaging studies support this global broadcasting model by showing that conscious perception involves increased communication between distant brain regions compared to unconscious processing. The prefrontal cortex, parietal cortex, and posterior cingulate cortex form a network that activates during conscious awareness of various stimuli, suggesting these regions contribute to the global workspace that enables conscious experience. The default mode network represents one of neuroscience's most intriguing discoveries, a set of brain regions that becomes highly active during rest and introspection. This network includes the medial prefrontal cortex, posterior cingulate cortex, angular gyrus, and hippocampus, regions that show decreased activity during focused attention on external tasks, but increased activity during self-referential thinking, autobiographical memory retrieval, and mental time travel. The default mode network appears to support the continuous stream of internal mental activity that characterizes the resting mind. When external demands are minimal, consciousness turns inward, engaging in what researchers call mind-wandering or stimulus-independent thought. These mental activities include remembering past events, imagining future scenarios, considering the thoughts and feelings of others, and reflecting on personal goals and concerns. Attention networks work in opposition to the default mode network, redirecting consciousness towards specific objects or tasks in the external environment. The executive attention network, centered in the frontal and parietal cortices, maintains focus on goal-relevant information while filtering out distractions. The alerting network, involving brainstem and frontal regions, maintains vigilant readiness to respond to important stimuli. The orienting network, including parietal and frontal areas, directs attention to specific locations in space or features of objects. These attention networks operate through both top-down and bottom-up mechanisms. Top-down attention involves voluntary control of consciousness, deliberately focusing awareness on chosen targets while suppressing irrelevant information. Bottom-up attention responds to salient stimuli that capture consciousness automatically, such as sudden sounds or movements that might signal important events requiring immediate response. Sleep represents a dramatically altered state of consciousness that reveals important principles about the neural basis of awareness. During non-rapid eye movement sleep, consciousness largely disappears as thalamic circuits generate slow oscillations that synchronize cortical activity and prevent the normal flow of information between brain regions. The global workspace effectively shuts down, leaving only local processing without the integration necessary for conscious experience. Rapid eye movement sleep restores some aspects of conscious-like activity, while maintaining disconnection from the external world. Dreams during REM sleep can involve vivid sensory experiences, emotions, and narratives, suggesting that consciousness can operate independently of external sensory input. The brain generates these experiences through internally driven activity that activates many of the same regions involved in waking consciousness. The brain's energy consumption patterns reveal important insights about the neural basis of consciousness. Despite representing only 2% of body weight, the brain consumes roughly 20% of the body's total energy production. Remarkably, the difference in energy consumption between wake and sleep states is relatively small, suggesting that much of the brain's metabolic activity supports unconscious processing rather than conscious awareness. The default mode network accounts for a significant portion of the brain's baseline energy consumption, remaining active, even during anesthesia, and some vegetative states. This finding suggests that maintaining the neural infrastructure for consciousness requires substantial metabolic resources, even when conscious experience itself is absent. Mind-wandering occurs during roughly 50% of waking hours, demonstrating that consciousness frequently disengages from immediate environmental demands to pursue internal mental activities. This spontaneous mental activity serves important functions, including memory consolidation, creative problem-solving, and planning for future events. However, excessive mind-wandering can also contribute to rumination and negative mood states. The quality of mind-wandering varies considerably between individuals and mental states. Some forms involve pleasant anticipation of future events or positive memories, while others focus on regrets, worries, or repetitive negative thoughts. The content and emotional tone of mind-wandering activity correlate with measures of well-being and mental health, suggesting that the default mode network's activity patterns influence psychological functioning. Meditation practices provide a unique window into the voluntary control of consciousness, demonstrating how sustained training can modify attention networks and default mode activity. Experienced meditators show reduced default mode network activity during both meditation and rest states, along with enhanced executive attention and emotional regulation abilities. These findings suggest that consciousness can be systematically trained and modified through contemplative practices. Different meditation techniques produce distinct patterns of brain activation, with focused attention practices strengthening executive control networks, while open monitoring approaches enhance metacognitive awareness and present moment focus. Loving kindness meditation activates networks involved in social cognition and emotional processing, demonstrating the specificity of contemplative training effects. The hard problem of consciousness, formulated by philosopher David Chalmers, distinguishes between the easy problems of explaining cognitive functions and the hard problem of explaining why there is subjective experience at all. While neuroscience has made significant progress in understanding the neural mechanisms of attention, memory, and perception, the question of why these processes should be accompanied by inner experience remains deeply puzzling. Various theories attempt to bridge the explanatory gap between neural activity and conscious experience. Integrated information theory proposes that consciousness corresponds to integrated information in neural networks, with higher levels of integration producing richer conscious experiences. Global workspace theory focuses on the broadcasting of information across brain networks as the key to consciousness. Predictive processing theories suggest that consciousness arises from the brain's continuous attempts to predict and explain sensory input. The study of consciousness raises profound questions about the nature of subjective experience and its relationship to physical processes. While neuroscience continues to identify the neural correlates and mechanisms associated with conscious states, the fundamental mystery of how matter gives rise to mind remains one of science's greatest challenges, inviting continued investigation into the most intimate aspects of human existence. Emotions and decision-making represent two of the brain's most interconnected functions, challenging the traditional view that rational thinking operates independently of feeling. Modern neuroscience reveals that emotions and cognition work together through intricate neural networks that evaluate options, weigh outcomes, and guide behavior in ways that promote survival and well-being. Far from being obstacles to clear thinking, emotions provide essential information that enables adaptive decision-making in complex environments. Emotional processing begins in the limbic system, an ancient collection of structures that evolved to detect threats, identify opportunities, and motivate appropriate responses. The amygdala serves as the brain's alarm system, rapidly evaluating sensory input for emotional significance and triggering physiological responses before conscious awareness even registers potential dangers. This almond-shaped structure can initiate fight-or-flight responses within milliseconds, preparing the body for action while alerting other brain regions to pay attention. The amygdala contains distinct nuclei that process different aspects of emotional experience. The lateral nucleus receives sensory input and forms associations between neutral stimuli and emotional outcomes through fear conditioning. The central nucleus orchestrates autonomic and hormonal responses, activating the sympathetic nervous system and triggering stress hormone release. The basolateral complex integrates emotional information with cognitive processing, influencing memory formation and decision-making. Emotional memories formed through amygdalar processing prove remarkably persistent and resistant to extinction. The neural pathways that encode fear associations remain permanently etched in the brain, explaining why phobias and traumatic memories can persist for decades. However, the amygdala also supports positive emotions like excitement and pleasure, particularly when processing rewarding stimuli that signal opportunities for gain. The prefrontal cortex provides sophisticated emotional regulation capabilities that distinguish human behavior from more primitive responses. The orbitofrontal cortex evaluates the expected outcomes of different actions, integrating emotional signals with cognitive assessments to guide decision-making. This region represents the value of different options and updates these representations based on changing circumstances and new information. The ventromedial prefrontal cortex integrates emotional and cognitive information to generate gut feelings and intuitive judgments about complex situations. Damage to this region produces profound deficits in real-world decision-making, despite preserved intellectual abilities, demonstrating the crucial role of emotional processing in practical reasoning. Patients with such damage often make disastrous personal and financial decisions while performing normally on traditional cognitive tests. The anterior cingulate cortex monitors conflicts between competing options and signals when additional cognitive control is needed. This region activates when people experience uncertainty, make errors, or encounter situations where automatic responses conflict with goals. The anterior cingulate helps resolve these conflicts by recruiting additional prefrontal resources and adjusting behavior based on feedback. Reward systems centered on dopamine-producing neurons in the ventral tegmental area drive motivation and goal-directed behavior. These neurons respond not to rewards themselves but to unexpected rewards and cues that predict future rewards. This prediction error signal enables learning about which actions and situations lead to positive outcomes, gradually shaping behavior toward adaptive choices. The nucleus accumbens, a key component of the reward circuit, integrates dopaminergic signals with input from the amygdala and prefrontal cortex to motivate approach behaviors toward rewarding stimuli. This region becomes hyperactive addiction, where drugs hijack normal reward processing and create pathological motivation toward substances, despite negative consequences. Different neurotransmitter systems contribute distinct aspects to emotional and decision-making processes. Serotonin influences mood, impulse control, and social behavior, with low levels associated with depression and aggression. Norepinephrine enhances attention and arousal during stressful situations, preparing the brain and body for rapid responses. GABA provides inhibitory control that prevents emotional responses from becoming overwhelming or inappropriate. The somatic marker hypothesis, developed by neuroscientist Antonio Damasio, proposes that bodily emotional responses influence decision-making through unconscious signals that guide choices toward favorable outcomes. According to this theory, the brain continuously generates predictions about the emotional consequences of different actions, with these somatic markers biasing decisions before conscious deliberation begins. Research supporting the somatic marker hypothesis shows that people with ventromedial prefrontal damage fail to develop anticipatory emotional responses when making risky decisions. They continue making disadvantageous choices even when they intellectually understand the negative consequences, suggesting that emotional signals normally guide behavior in ways that transcend conscious reasoning. Social brain networks enable the recognition and understanding of others' emotions, facilitating cooperation and social coordination. The superior temporal sulcus processes biological motion and facial expressions, while the temporoparietal junction enables perspective taking and theory of mind. Mirror neuron systems in the premotor and parietal cortices activate both when performing actions and observing others perform similar actions, providing a neural basis for empathy and emotional contagion. The stress response system demonstrates the intimate connection between emotion and physiological regulation. The hypothalamic pituitary adrenal axis responds to psychological stress by releasing cortisol, which mobilizes energy resources while suppressing non-essential functions like immunity and digestion. Chronic stress activation can impair memory, decision-making, and physical health, illustrating how emotional states influence cognitive function and bodily well-being. Emotional regulation strategies vary in their effectiveness and neural mechanisms. Cognitive reappraisal involves reinterpreting situations to change their emotional impact, recruiting prefrontal regions that modulate amygdala responses. Suppression attempts to inhibit emotional expression without changing the underlying feeling, often proving counterproductive and increasing physiological stress. Mindfulness approaches encourage accepting emotions without judgment while maintaining cognitive flexibility. The development of emotional and decision-making capabilities follows a protracted course that extends through adolescence. The limbic system matures earlier than prefrontal control regions, creating a period of heightened emotional reactivity combined with limited regulatory abilities. This developmental imbalance explains the increased risk-taking and emotional volatility characteristic of adolescence. Cultural factors significantly influence both emotional expression and decision-making processes. Different societies emphasize distinct emotional values, with some cultures prioritizing individual emotional expression while others emphasize collective harmony and emotional restraint. These cultural differences produce variations in neural activation patterns during emotional processing, demonstrating the role of experience in shaping brain function. Individual differences in emotional processing reflect both genetic factors and learned patterns developed through experience. Some people show heightened amygdala reactivity that predisposes them to anxiety and emotional volatility, while others demonstrate enhanced prefrontal control that facilitates emotional regulation. Understanding these differences can inform therapeutic approaches and help individuals develop more effective emotional management strategies. The integration of emotional and cognitive processes enables the flexibility and adaptability that characterize human behavior. Rather than viewing emotion and reason as opposing forces, modern neuroscience reveals them as complementary systems that work together to navigate complex social and physical environments, making decisions that balance immediate needs with long-term goals, while maintaining the social relationships essential for human flourishing. The human brain embarks on a remarkable journey across the lifespan, transforming from a collection of undifferentiated cells into the most complex structure in the known universe, then gradually adapting and changing as decades pass. This developmental odyssey spans from the earliest moments of conception through the wisdom of old age, revealing how genetic programs and environmental influences collaborate to shape the neural architecture that defines each individual's unique cognitive landscape. Brain development begins in the third week after conception, when the neural tube forms from a simple sheet of cells. This primitive structure gives rise to the entire central nervous system through an intricate choreography of cellular division, migration, and differentiation. Neural stem cells proliferate at extraordinary rates, generating neurons at peak production rates of over 250,000 per minute during certain stages of prenatal development. Cell migration represents one of development's most remarkable processes, as newly formed neurons travel vast distances in cellular terms to reach their final destinations. Radial glial cells serve as scaffolding for this migration, extending processes from the brain's interior to its surface, along which neurons climb like mountain climbers following guide ropes. Chemical signals attract neurons to appropriate regions, while repulsive cues prevent them from settling in incorrect locations. The formation of synaptic connections follows principles of both genetic programming and activity-dependent refinement. Initial connections form through molecular recognition systems that guide axons to appropriate targets, but these early circuits undergo extensive modification based on patterns of neural activity. Synapses that participate in correlated activity strengthen through Hebbian learning mechanisms, while unused connections weaken and eventually disappear through a process called synaptic pruning. Childhood brain development involves a fascinating interplay between synapse formation and elimination. During early childhood, the brain produces synapses at rates far exceeding adult levels, creating a dense network of connections that provides maximal plasticity for learning. However, this exuberant connectivity must be refined through experience-dependent pruning that eliminates unnecessary connections, while strengthening those that prove useful for navigating the child's specific environment. Different brain regions follow distinct developmental timetables that reflect their functional priorities. Sensory and motor areas mature relatively early, enabling basic perceptual and movement capabilities during infancy. Language regions undergo rapid development during the preschool years, coinciding with the explosive growth of vocabulary and grammatical abilities. Executive control regions in the prefrontal cortex mature last, not reaching full development until the mid-20s. Myelination proceeds gradually throughout childhood and adolescence, with some white matter tracts continuing to mature into the fourth decade of life. The progressive addition of myelin sheaths increases transmission speed and enables more sophisticated cognitive abilities that depend on rapid communication between distant brain regions. The corpus callosum connecting the two hemispheres continues myelinating well into adolescence, potentially explaining developmental changes in interhemispheric coordination. Adolescent brain development involves particularly dramatic changes in regions involved in emotional processing, social cognition, and risk assessment. The limbic system matures earlier than prefrontal control regions, creating a temporary imbalance between emotional reactivity and regulatory capabilities. This developmental mismatch may explain the increased risk-taking, emotional volatility, and peer influence susceptibility characteristic of adolescence. The adolescent brain shows heightened sensitivity to rewards, particularly social rewards like peer approval and novel experiences. Dopamine systems in the nucleus accumbens become hyper-responsive during adolescence, making teenagers particularly susceptible to the rewarding effects of risky behaviors, while their prefrontal cortex remains immature and less capable of inhibiting impulsive choices. Adult neuroplasticity reveals that the brain retains remarkable capacity for change throughout life, contradicting earlier beliefs about fixed neural architecture after critical periods. Experience can modify synaptic connections, alter gene expression, and even generate new neurons in certain brain regions. This ongoing plasticity enables lifelong learning and adaptation while providing mechanisms for recovery following brain injury. The adult hippocampus represents one of the few brain regions where new neurons continue to be generated throughout life. Adult neurogenesis contributes to learning and memory functions while potentially playing roles in mood regulation and stress resilience. Environmental factors like exercise, enriched social environments, and cognitive challenges can enhance neurogenesis rates, while stress and depression tend to suppress the production of new neurons. Learning-induced plasticity demonstrates how experience reshapes neural architecture throughout adulthood. Intensive training in specific skills produces measurable changes in brain structure, including increased gray matter volume in relevant regions and enhanced white matter integrity in connecting pathways. Musicians show enlarged motor and auditory areas. Taxi drivers develop enlarged posterior hippocampi for spatial navigation, and jugglers demonstrate increased gray matter in visual motor coordination regions. Brain aging involves complex changes that affect different regions and functions at varying rates. Some cognitive abilities like crystallized intelligence and vocabulary remain stable or even improve with age, while processing speed and working memory show gradual decline beginning in early adulthood. These differential aging patterns reflect the varying vulnerability of different neural systems to age-related changes. The aging brain undergoes structural modifications including modest volume reductions, decreased white matter integrity, and changes in neurotransmitter systems. However, These changes occur gradually and vary tremendously between individuals. Many older adults maintain excellent cognitive function despite measurable brain changes, demonstrating the brain's capacity to compensate for age-related alterations through adaptive mechanisms. Cognitive reserve represents the brain's ability to maintain function despite age-related changes or pathological processes. Individuals with higher educational attainment, complex occupational histories, and active social engagement often show greater resilience to cognitive decline. This protective effect may result from more efficient neural networks, enhanced compensatory mechanisms, or greater structural brain reserves developed through lifelong mental stimulation. The aging brain demonstrates remarkable compensatory abilities, often recruiting additional neural resources to maintain performance levels. Older adults frequently show bilateral activation patterns during cognitive tasks that engage only one hemisphere in younger individuals. This age-related increase in bilateral activation may represent a successful adaptation that helps maintain cognitive abilities despite declining efficiency in specific brain regions. Environmental factors profoundly influence brain development and aging across the lifespan. Enriched environments with cognitive challenges, social interaction, and physical activity promote neural health and plasticity. Conversely, chronic stress, social isolation, and sedentary lifestyles can accelerate cognitive decline and increase vulnerability to neurodegenerative diseases. Physical exercise provides particularly robust benefits for brain health across all life stages. Aerobic exercise enhances neurogenesis, promotes the production of growth factors like brain-derived neurotrophic factor, and improves vascular health that supports neural function. Regular physical activity during midlife appears especially protective against later cognitive decline and dementia risk. The concept of successful aging encompasses not merely the absence of disease, but the maintenance of cognitive abilities and functional independence throughout later life. Research identifying factors that promote successful brain aging reveals the importance of lifelong learning, social engagement, physical activity, and stress management in preserving neural function and supporting quality of life across the entire lifespan. Neural efficiency emerges as a hallmark of wisdom, with experienced older adults often demonstrating more selective and strategic use of cognitive resources compared to younger individuals. Rather than simply declining, the aging brain may develop sophisticated strategies for conserving energy while maintaining performance, suggesting that neural maturation continues throughout life in ways that transcend simple measures of processing speed or memory capacity. The brain's remarkable complexity makes it vulnerable to a wide range of disorders that can disrupt cognition, emotion, movement, and consciousness itself. These conditions illuminate the intricate relationships between neural structure and function, while revealing the brain's extraordinary capacity for adaptation and recovery. Understanding neurological and psychiatric disorders provides crucial insights into normal brain function, while highlighting the resilience mechanisms that can restore abilities even after significant neural damage. Stroke represents one of the most common and devastating brain disorders, occurring when blood flow to brain tissue becomes interrupted either through blockage or rupture of blood vessels. Ischemic strokes, caused by clots that obstruct cerebral arteries, account for roughly 85% of all strokes, while hemorrhagic strokes result from bleeding within brain tissue. The location and extent of stroke damage determine the specific deficits that emerge, from motor paralysis and speech difficulties to memory loss and personality changes. The brain's response to stroke demonstrates remarkable plasticity as undamaged regions attempt to compensate for lost functions. Immediately following stroke, neural circuits adjacent to damaged areas often become hyperexcitable as they lose normal inhibitory input. Over weeks and months, these surviving regions can reorganize their connections and take on new functions, enabling partial recovery of abilities that initially appeared permanently lost. Rehabilitation following stroke capitalizes on this neural plasticity through intensive training programs that encourage the formation of new neural pathways. Constraint-induced movement therapy forces patients to use affected limbs by restraining unaffected ones, promoting reorganization of motor cortex. Speech therapy similarly encourages language recovery by engaging remaining language networks and establishing alternative communication pathways. Neurodegenerative diseases involve the progressive loss of neurons and their connections, typically affecting specific brain regions in characteristic patterns. Alzheimer's disease, the most common form of dementia, begins with the accumulation of abnormal proteins called amyloid plaques and tau tangles that disrupt cellular function and eventually cause neuronal death. The disease typically starts in the entorhinal cortex and hippocampus, explaining why memory loss often represents the earliest symptom. Parkinson's disease results from the death of dopamine-producing neurons in the substantia nigra, leading to the characteristic motor symptoms of tremor, rigidity, and bradykinesia. The loss of dopaminergic input to the basal ganglia disrupts the normal flow of movement control signals, making it difficult to initiate and coordinate voluntary movements. However, Parkinson's also affects non-motor functions, as the disease progresses to involve other neurotransmitter systems. Huntington's disease demonstrates how genetic mutations can cause selective neuronal variability. The expansion of CAG repeats in the hunting gene leads to progressive degeneration of medium spiny neurons in the striatum, producing involuntary movements and cognitive decline. This autosomal dominant disorder illustrates how single gene defects can have widespread consequences for brain function and behavior. Mental health conditions increasingly appear to involve disruptions in neural circuits rather than discrete brain lesions. Major depression involves alterations in neurotransmitter systems, particularly serotonin, norepinephrine, and dopamine, along with structural changes in regions like the hippocampus and prefrontal cortex. The default mode network shows altered connectivity patterns in depression, potentially explaining the rumination and negative self-focus characteristic of this condition. Schizophrenia reflects complex interactions between genetic vulnerability and environmental factors that affect neurodevelopment. The disorder involves widespread alterations in brain connectivity, neurotransmitter function, and cortical organization that emerge during late adolescence or early adulthood. Positive symptoms like hallucinations and delusions may result from excessive dopamine activity, while negative symptoms and cognitive deficits involve dysfunction in prefrontal circuits. Anxiety disorders demonstrate how normal fear and stress responses can become dysregulated, leading to persistent worry and avoidance behaviors. The amygdala shows heightened reactivity in anxiety disorders, while prefrontal regions responsible for emotional regulation may show decreased activity. These circuit imbalances create a state where threat detection systems remain chronically activated even in safe environments. Traumatic brain injury can cause both immediate and long-term consequences depending on the mechanism and severity of damage. Diffuse axonal injury, common in motor vehicle accidents, involves widespread damage to white matter tracts that connect different brain regions. Even mild traumatic brain injuries can produce lasting cognitive and emotional effects through disruption of neural networks rather than focal brain lesions. The brain's response to traumatic injury involves complex cascades of cellular and molecular events that can either promote recovery or cause additional damage. Inflammation, while initially protective, can become destructive if prolonged. Excitotoxicity occurs when damaged neurons release excessive glutamate, causing further cellular death in surrounding tissue. Understanding these secondary injury mechanisms has led to therapeutic approaches aimed at neuroprotection and recovery enhancement. Seizure disorders reveal how disruptions in the delicate balance between excitation and inhibition can produce dramatic alterations in consciousness and behavior. Epileptic seizures result from synchronous firing of large groups of neurons, creating electrical storms that can spread throughout the brain. Different seizure types reflect the brain regions involved and the pattern of abnormal electrical activity. Modern treatments for seizure disorders include both medications that restore excitatory-inhibitory balance and surgical approaches that remove or disconnect seizure-generating brain regions. Deep brain stimulation and responsive neurostimulation represent newer approaches that can interrupt seizure activity before it spreads. These interventions demonstrate how understanding neural circuit dysfunction can lead to targeted therapeutic strategies. Autism spectrum disorders involve differences in neural connectivity and information processing that affect social communication and behavioral flexibility. Research suggests that autism may involve altered patterns of synaptic pruning during development, leading to differences in local versus long-range connectivity. These connectivity differences may explain both the challenges and strengths associated with autism, including enhanced attention to detail and pattern recognition abilities. Attention deficit hyperactivity disorder reflects differences in executive attention networks and reward processing systems. Neuroimaging studies reveal altered activity in prefrontal regions responsible for impulse control and working memory, along with differences in dopaminergic circuits involved in motivation and reward processing. These findings have informed both medication approaches and behavioral interventions for ADHD. The brain's capacity for adaptation extends beyond recovery from injury to include compensation for congenital differences and progressive diseases. Individuals born blind show enhanced development of auditory and tactile processing regions, demonstrating how sensory deprivation can lead to expanded capabilities in remaining modalities. Similarly, people with dyslexia often develop enhanced visual spatial abilities and creative problem-solving skills. Modern therapeutic approaches increasingly target the brain's plasticity mechanisms to promote recovery and adaptation. Transcranial magnetic stimulation can enhance or suppress activity in specific brain regions, potentially facilitating rehabilitation. Brain-computer interfaces offer hope for individuals with severe motor impairments by allowing direct neural control of external devices. Cognitive training programs aim to enhance specific abilities through targeted practice that promotes beneficial neural changes. Pharmacological interventions for brain disorders continue to evolve as understanding of neurotransmitter systems and neural circuits advances. New antidepressants target specific receptor subtypes or multiple neurotransmitter systems simultaneously. Medications for neurodegenerative diseases increasingly focus on disease-modifying approaches rather than just symptom management. Personalized medicine approaches consider genetic factors that influence drug metabolism and response. The future of brain disorder treatment lies in early detection, prevention, and interventions that harness the brain's natural recovery mechanisms. Biomarker research aims to identify brain changes before symptoms appear, enabling preventive interventions. Stem cell therapies and tissue engineering approaches may eventually replace damaged neurons and restore lost functions. Gene therapy techniques could potentially correct genetic defects that cause inherited brain disorders. Understanding brain disorders ultimately reveals both the vulnerability and resilience of neural systems, demonstrating how the same plasticity mechanisms that enable learning and adaptation can also facilitate recovery from injury and compensation for dysfunction, offering hope for the millions of individuals affected by neurological and psychiatric conditions. The human brain operates not as a collection of isolated modules but as an integrated network of networks where billions of neurons coordinate their activity across vast distances to generate the seamless unity of thought, perception, and action. This connected architecture represents one of nature's most sophisticated achievements in distributed information processing, enabling complex behaviors to emerge from the coordinated activity of relatively simple cellular components working together across multiple spatial and temporal scales. Large-scale brain networks organize themselves into distinct functional systems that can be identified through their patterns of synchronized activity. The default mode network activates during rest and introspection, connecting regions involved in self-referential thinking and autobiographical memory. The executive control network engages during demanding cognitive tasks, linking prefrontal and parietal areas that direct attention and manage working memory. The salience network monitors both internal thoughts and external events, determining which information deserves conscious attention and cognitive resources. These networks operate through dynamic patterns of communication that change depending on behavioral demands and cognitive states. During focused attention tasks, the executive control network becomes highly active while the default mode network shows decreased activation, reflecting the brain's ability to allocate resources efficiently. The salience network serves as a switch between internal and external focus, helping coordinate transitions between different cognitive modes. The connectome represents the complete map of neural connections within the brain, analogous to the genome's comprehensive description of genetic information. Modern neuroimaging techniques can trace white matter pathways throughout the living brain, revealing the structural highways that enable communication between distant regions. These anatomical connections constrain but do not completely determine functional connectivity, as the strength of communication between regions can vary dramatically depending on current brain states and activities. Structural connectivity follows organizing principles that balance competing demands for efficiency and flexibility. The brain minimizes wiring costs by placing frequently communicating regions close together, while maintaining long-range connections that enable global integration. This architecture creates a small world network, where most regions can communicate with any other region through just a few intermediate steps. Similar to the 6 degrees of separation phenomenon in social networks, hub regions occupy central positions in the brain's connectivity matrix, receiving and sending connections to numerous other areas. The posterior cingulate cortex, precuneus, and medial prefrontal cortex serve as major hubs in the default mode network, while the anterior insular and dorsal anterior cingulate cortex function as key nodes in the salience network. Damage to these highly connected regions often produces widespread functional disruptions that exceed what would be expected from their anatomical size alone. Neural oscillations provide temporal coordination mechanisms that enable distant brain regions to communicate effectively, despite the delays inherent in long-range connections. Different frequency bands serve distinct functional roles. Gamma rhythms support local processing and binding. Beta oscillations coordinate motor control. Alpha waves regulate attention and arousal, and delta rhythms characterize deep sleep states. These rhythmic patterns create temporal windows during which neurons can communicate most effectively. Synchronization between brain regions occurs when their neural oscillations align in phase, creating moments of enhanced communication efficiency. This mechanism allows the brain to dynamically configure itself into different functional networks depending on task demands. During visual attention tasks, synchronization between visual cortex and frontal attention areas enhances processing of relevant information while suppressing irrelevant inputs. The brain's network organization exhibits hierarchical structure, with local circuits embedded within larger regional networks that in turn participate in global brain-wide systems. Primary sensory areas form local processing modules that feed into association regions, which then connect to executive control networks. This hierarchical organization enables the brain to process information at multiple scales simultaneously, from detecting simple features to integrating complex concepts. Information integration across brain regions enables the binding of distributed neural representations into unified perceptual experiences and coherent thoughts. The binding problem in neuroscience addresses how the brain combines information processed in separate locations into seamless conscious experiences. Current theories propose that temporal synchronization and convergent connectivity patterns provide mechanisms for integrating distributed neural activity. Network efficiency reflects the brain's ability to transfer information with minimal energy expenditure and processing delays. Highly efficient networks maintain short path lengths between nodes while using relatively few connections, maximizing information flow while minimizing metabolic costs. Individual differences in network efficiency correlate with cognitive abilities and may explain variations in intellectual performance across the population. The brain demonstrates remarkable adaptability in its network organization, with connectivity patterns changing in response to learning, development, and injury. Practice strengthens connections between regions involved in specific skills, while disuse weakens unnecessary pathways. This plastic reorganization enables the brain to optimize its network structure based on individual experience and environmental demands. Brain networks exhibit complex dynamics that operate far from equilibrium, constantly fluctuating between different states while maintaining overall stability. These dynamics enable rapid transitions between cognitive states while preventing the system from becoming locked into rigid patterns. Critical dynamics at the edge of chaos may optimize the brain's computational capabilities by maximizing both stability and flexibility. The emergence of complex behaviors from network interactions demonstrates how simple local rules can generate sophisticated global phenomena. Individual neurons follow relatively straightforward input-output relationships, yet their collective interactions produce the full spectrum of human cognition and behavior. This emergence represents one of the most profound examples of how complexity can arise from simplicity in biological systems. Modern network neuroscience employs mathematical tools from graph theory to quantify brain organization and identify disruptions associated with neurological and psychiatric disorders. Depression involves altered connectivity within the default mode network, while schizophrenia shows disrupted long-range connections and altered small-world properties. These network-level changes provide new perspectives on mental illness that complement traditional approaches focused on individual brain regions. Computational models of brain networks help researchers understand how network structure influences function and predict how disruptions might affect behavior. These models reveal that the brain's architecture optimizes information processing while remaining robust to damage, explaining how the brain can maintain function despite age-related changes or focal injuries. Network modeling also suggests therapeutic targets for disorders involving connectivity disruptions. The study of brain networks across species reveals both conserved principles and evolutionary innovations in neural organization. Mammalian brains share fundamental network motifs, while differing in the specific regions that serve as major hubs. Human brains show expanded association areas and enhanced long-range connectivity that may underlie our species' unique cognitive capabilities. Future research directions in network neuroscience include developing better methods for measuring dynamic connectivity, understanding how networks develop across the lifespan, and translating network findings into clinical applications. Brain stimulation techniques may eventually target specific network properties to enhance cognitive function or treat psychiatric disorders. The integration of network approaches with molecular and cellular neuroscience promises deeper understanding of how genetic and environmental factors shape brain connectivity. The connected brain represents a paradigm shift from viewing the nervous system as a collection of specialized modules toward understanding it as an integrated complex system. This perspective reveals how the remarkable capabilities of human cognition emerge from the coordinated activity of neural networks that span the entire brain, creating the unified conscious experience that defines our existence while maintaining the flexibility to adapt and learn throughout life. Our journey through the neural landscapes of the human brain brings us full circle to a profound realization. The very organ we have explored continues its quiet work even as consciousness fades into sleep. As you listen to these final words, billions of neurons maintain their ceaseless coordination, preserving the essential rhythms of life while preparing for the restorative processes that unfold in dreams. The brain we have discovered together represents both humanity's greatest achievement and its deepest mystery. From the ancient brainstem structures that emerged hundreds of millions of years ago, to the sophisticated cortical networks that enable abstract thought, this three-pound universe contains the biological foundation for everything that makes us human. Yet for all our scientific progress, vast territories remain unexplored, fundamental questions unanswered. How does the firing of neurons give rise to the felt experience of consciousness? What mechanisms enable the seamless integrity of memory, emotion, and reasoning that characterizes human cognition? How do billions of individual cells coordinate their activity to produce the unified sense of self that persists across decades of change? These mysteries continue to challenge our understanding while inspiring new generations of researchers to probe deeper into the brain's secrets. As sleep approaches, the same neural networks we have explored will orchestrate the transition from waking awareness to restorative slumber. The default mode network will guide your mind through its final wanderings before consciousness dissolves into the rhythmic oscillations of sleep. In the quiet darkness ahead, your brain will continue its essential work, consolidating memories, clearing metabolic waste, and preparing for another day of the endless miracle we call human experience. Rest well, knowing that this remarkable organ will faithfully maintain its vigil through the night.